Okay, as promised, here in the third lecture for Chapter 9, we're going to start moving into the microanatomy of your muscle fibers, your muscle cells. And we're thinking about our skeletal muscle fibers here in Chapter 9 for the most part. All right, just some information for you about your skeletal muscle fibers. Again, they look like cylinders, as we've already seen, and they can be 10 to 100 micrometers in diameter. And see, a micrometer is 1/1,000,000th of a meter. 100 micrometers, maybe you've seen a metric ruler before, so you know how thin 1 millimeter is on a metric ruler. Not much wider than a piece of paper. And then 1/10 of that would be 100 micrometers. So they're very, they're microscopic, of course, at that level. But then look how long they can be, 30 centimeters long. So that's like a foot long in some cases. An individual cell can be that long. If you think about some of your skeletal muscles, the individual muscles are pretty large, and so individual cells within those muscles can, in some cases, be over 10 inches in length. So that's very unusual for individual cells in your body. Multinucleated, as we've already mentioned, and the nuclei tend to be squished over along the edges rather than being in the center of the cell. They contain many mitochondria. And do you remember what mitochondria are used for back from Unit 1? Those are organelles inside the cells, and their main job is ATP production. Mitochondria, those are the locations where most of the chemical reactions for cellular respiration take place, in the mitochondria. So it should not be a surprise to you that since these cells contract, that takes energy. Well, guess what they need to make a lot of? They need lots and lots of ATP. Another thing that your skeletal muscle fibers have, glycosomes are little storage areas for glycogen. Glycogen is a starch that your skeletal muscle cells make, and they're basically glucose chains. They're polysaccharides. So since glucose is a favorite energy source for cells, your skeletal muscles need lots of energy. So they'll actually store, when you have excess glucose available, they'll actually link glucose together in chains to make glycogen, and they'll store that on the inside so that they can use it when they're having to do lots of contractions. There's also a type of protein in your skeletal muscles, let me erase all that, called myoglobin, which stores O2, oxygen gas. What do we need O2 for? Good old cellular respiration. Why do we need to do cellular respiration? Because cellular respiration is how we make ATP. So myoglobin is actually a protein that harvests oxygen gas out of the blood and keeps it inside your skeletal muscle fibers. So they're basically oxygen gas hoarders for the most part, because they need it. They've got to have it to power muscle contractions. Myoglobin is related to hemoglobin. That's a protein in your red blood cells that binds oxygen gas and transports that around your body. So if you've heard of hemoglobin before, myoglobin is related to hemoglobin. Myo, by the way, that prefix, myo, tends to refer to muscles. So when you see that, myo often refers to that. All right, also within a skeletal muscle fiber, you're going to have structures called myofibrils. We saw a little bit about those on the last lecture video. The sarcoplasmic reticulum, we'll talk about what that is, and structures called T-tubules. So we'll see what those are as well. I do want to point out to you that within this chapter, your textbook publishers have a lot of excellent beautifully done animations. And many of the things we're talking about here, I'm going to do my best to try to explain them to you here off of these two-dimensional slides. But to really get a good mental picture for what's going on, take advantage of the wonderful animations that your textbook publishers have created. Because I can sit here all day and draw pictures and arrows and scribble all over these slides, but when you see it animated, that's when it really makes sense. All right, so here we are zooming in on a portion of a single skeletal muscle fiber. And of course, this little transparent thing they're peeling off there is supposed to be the plasma membrane or the cell membrane that they're cutting open. The cell membrane or the plasma membrane of a muscle fiber has a special name, sarcolemma. That is the plasma membrane of a skeletal muscle fiber. So get used to hearing that term. Sarcolemma is referring to the plasma membrane of a skeletal muscle fiber. Notice here on the diagram they're highlighting for you those little things that look like orange sausages in there. Those are the mitochondria. Those are the ATP generating organelles. And then within the muscle fiber, these things that kind of look like pink candy cigarettes that are being pulled out, those are called myofibrils. So you can see the bulk of the space inside a muscle fiber consists of a whole bunch of myofibrils. And again, what those are not cells. This is a cell. When you get to the myofibril layer, you're looking at a part of an individual cell, and those are bundles of proteins that are involved in muscle contraction. Those are also the parts that have the striped look. So when you look at them under a microscope, that's where you're getting your striations or that striped appearance. All right, so now let's zoom in and take a closer look at a myofibril. So here you're looking at one of those protein cylinders that's involved in muscle contraction. And just because we need even more terminology from the muscle specialists, a myofibril is a chain of sarcomeres. So a sarcomere is the basic repeating unit of these myofibrils. So myofibrils, again, consist of different proteins that are bundled together and they're involved in muscle contraction. But then those myofibrils can be chopped up into individual sarcomeres. So sarcomere is the structural unit. All right, an individual sarcomere, so here on this little chunk of a myofibril we're looking at, everything from here over to here is a sarcomere. All right, and then you're going to have another one going from here over to here. They've cut that one off in half. And you're going to have another one going from here to over here. So a whole myofibril, which may extend for a very long length, is going to be a bunch of repeating units of these things right here that we call sarcomeres. Now, if you're reading along in your textbook, the textbook is going to talk about Z discs and H zones and A bands and I bands, and we're not going to get into all that. M lines on there as well. I'll talk a little bit about some of those things, but don't get stressed out if you're reading in the textbook about the meaning of all of those different things. I think that's going overboard. There's already enough terminology here, and we don't need to get into that. That's some of the traditional skeletal muscle terminology that was generated a long time ago when people were trying to figure out what all those striations and stripes were. As it turns out, all that striping you see, and you can see the striped appearance there on the myofibril, is based on the types of proteins that are in there, which are involved in muscle contraction. The two main ones that you have in here are called thin filaments and thick filaments. Thin filaments are built from a protein called actin, and thick filaments are built from a protein called myosin. Filament is another term for like a thread. So you have a thread-like structure. That's where filament comes from. Like the filament in a light bulb, for example, if you've heard that before. All right, so as I mentioned over there on that other diagram, the thick filaments on that diagram are composed of a protein called myosin that's very important for muscle contraction. And then the thin ones are built from a protein called actin, and we'll see more about those shortly. Back on that other diagram, the Z discs, which those form the borders between sarcomeres, the Z discs, they kind of have that zigzag shape on the diagram. So what you have there, those are built from proteins that help anchor. You've got thin filaments that are connected to those, the Z discs. So there's one of your Z discs right there. That zigzag line. There's another one over here. These would be thin filaments. So they're anchored into those Z discs. And also those Z discs are the borders between one sarcomere and another. And what we're going to be learning about is that your actin and myosin filaments, so your myosin thicker filaments would kind of look like this in the center of the sarcomere there, being overlapped by the thin ones. And basically what happens when a skeletal muscle contracts is within your muscle cells, you have these sarcomeres where the actins and the myosins overlap each other like this. They basically walk across each other. And as that happens, the sarcomere does this. It gets shorter. Now we just said that myofibrils are a chain of a bunch of sarcomeres. So if all the sarcomeres in that myofibril are doing this, the whole myofibril is going to contract. And then we said that a muscle cell is a bundle of a whole bunch of those myofibrils. So within that cell, if you have a bunch of all the myofibrils are doing this at one time, then that whole cell is going to contract. Now think about a whole muscle like your biceps brachii muscle. It's going to be built from many muscle cells. And so within that muscle, if you have individual cells that are doing this, that's going to make the whole muscle contract. And so just think about that a little bit. It's hard when you're first learning about this to keep all these different levels of organization of these muscles and individual muscle cells straight. It just takes some practice and thinking through it to get used to it. And here's another look at that. Okay, so we're zooming in here now deep inside, and we're looking inside an individual sarcomere, and we're looking at those thin and those thick filaments. All right, so here are your, let me flip back over to my thinner pen. All right, there's a thin filament, and then in red over here, there's a thick filament. The thick filaments are built from a protein called myosin. So get used to hearing about that. An individual myosin protein kind of looks like this. It almost looks like a two-headed snake, or like a two heads that are attached onto two snakes that are twisted around each other maybe. And then to build a thick filament, you take a whole bunch of these myosins and bundle them together like you see here. And so that's why you see those heads, those myosin heads, scattered all along the thick filament there. A thin filament is different. A thin filament is built mainly from a protein called actin, which kind of looks like these blue olives, like a blue pitted olive or something that you see there. Those are proteins. Those are individual protein molecules. They kind of look like blue beads or whatever. And a thin filament has two chains of those actin bead-like proteins that are strung together and wrapped around each other, kind of twisted around each other. Then you've also got a couple of other proteins there, one called tropomyosin and the other one called troponin. So we'll be hearing about those a whole lot more as we move through this unit. Yeah, and here's a zoom in on the thin filament, the actin filament, since you've got more than one type of protein there so that we can take a closer look. So again, in blue there, you can see a single actin filament is really built from, or a single thin filament, is 2 strings of actin protein beads that are linked together. And then this protein here that kind of looks like a, let me change my pen color for a second. Right in here, that protein that kind of snakes its way across the surface of one of your actin strings, and then you've got another one over here. That's called tropomyosin. So tropomyosin is a long, stringy protein. Remember how back in Unit 1 I warned you that proteins are very, very diverse, and here you're seeing some examples of this. Proteins are all chains of amino acids, but the way those amino acids are put together and how many of them there are and which ones are used, you have 20 different types, dictates their shapes and also their function. So you can see proteins can have very different shapes. Actin looks like a bead. Tropomyosin looks like a ribbon, basically. All right, and then finally you have a third type of protein in these thin filaments, like right there and right there and right there and right there. Those are called troponin proteins. And so all 3 of those are going to have an important part in the story of muscle contraction. Again, what we're going to be learning about, for muscles to contract, you've got to have actin thin filaments and those myosin thick filaments. They come together, and then what's going to happen is they're going to slide across each other. And that's what makes a skeletal muscle contract. And that sounds really simple, but it's actually a pretty complex story for how all that works, and that's what we're going to be learning about most of the time here in the first part of Chapter 9. All right, a couple of other things to talk about inside a skeletal muscle fiber. So the red bundles you see there, those are parts of myofibrils that have been cut open. They're showing you over here. Let me change my pen color so it's more visible. All right, so you're looking at like a chunk inside a muscle cell or a muscle fiber. There's your sarcolemma, that's the cell membrane. There's some mitochondria in there as well. The structures they have colored in blue there and all over that kind of look webby, that is called the SR, or the sarcoplasmic reticulum. That may remind you of the endoplasmic reticulum from back when we did a little bit about cell structure back in Unit 1. And that's what it is. The sarcoplasmic reticulum is a membrane-covered, webby-looking compartment that is really smooth endoplasmic reticulum, if you remember that from Unit 1. But in a muscle cell, muscle fiber, it's called sarcoplasmic reticulum. Sarco, that prefix a lot of times is referring to muscle fibers. All right, so you have those. And then if you notice on here also, kind of running in a perpendicular direction and looping around myofibrils right there and over here. Each of those is called a T-tubule, or a transverse tubule would be another name for those. T-tubule or transverse tubule. So they run across or perpendicular to the sarcoplasmic reticulum compartments. These are membrane-covered compartments. And one of their main functions is going to be storage of calcium ions, as we'll see as we move through the units. Then another term you should be familiar with. Notice that the sarcoplasmic reticulum, let me erase that for a second. Notice how the sarcoplasmic reticulum gets a little wider, and there are parts of it that run right up next to those T-tubules like that. Those are called the terminal cisternae. I guess I just wrote over what I was writing there. Those are called the terminal cisternae of the sarcoplasmic reticulum, where they're running right up against those transverse tubules. All right, so we'll be hearing about those parts of your skeletal muscle fibers as we move through the unit as well. Okay, so here's, on these slides, here are some things that are just written out that I just mentioned when we were going over the diagram. So there's your SR again, is really just a bunch of smooth endoplasmic reticulum that looks webby and it surrounds each myofibril. And then you've got the terminal cisternae, those are, or cisternae, those are up against, those are portions of the SR that are adjacent to those T tubules. And as we're going to see, their main function, controlling calcium ion levels intracellularly, inside the cytosol, the fluid inside those muscle fibers, which winds up being extremely important for muscle contraction. Remember how we said calcium homeostasis, very important? Well, now you're really going to see an example. Calcium ions are critical for proper skeletal muscle contractions, actually the contractions of any of the different types of muscle tissues that you have. Calcium is also important for the function of your nerve cells. Your nervous system would not be able to function without proper calcium levels either. All right, and then the T tubules again. Notice on here, we saw what those looked like on the skeletal muscle fiber diagram, but notice here they're continuous with the sarcolemma. So they're little, what they are, all right, so if this is the sarcolemma again is the cell membrane, the plasma membrane of a muscle fiber. A T tubule is basically an invagination where it goes down inside into the interior of the muscle fiber and it kind of wraps around myofibrils that you have inside there. So it's like an invagination, an indentation of the cell membrane or the sarcolemma, and it goes inside the cell. So that'll be important a little bit later as we go through our muscle fiber contraction story. So just keep that in mind. All right, so that gives us an overview of the critical parts inside a skeletal muscle fiber. You may want to, before you move on to the next lecture, spend some time practicing with that new terminology so that you're familiar with what those words mean, because if you don't, as we keep moving through these different lectures, I'm very quickly going to sound like I'm just up here going wah, 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 like the teacher on Charlie Brown if you don't learn that new terminology. So try to spend some time doing that before you move on to these additional lectures.